All right, so now we are going to take this diploid cell that we just talked about with our homologous chromosome example, and we're going to send it through mitosis. So again, at this point, we have 4 chromosomes. We have 2 chromosome number 1s, 2 chromosome number 2s, so again, 2 homologous pairs, and now we're going to go through mitosis. Okay, so if we start first, of course, with interphase, the cell is going to go through G1 to begin with. Main thing that's happening here is this cell is just physically getting bigger. Okay? The chromosomes really have not changed. We still have chromosome 1 and 2 from the egg or sperm, and chromosome 1 and 2 from the egg. So we still have our 4 chromosomes. The cell's getting bigger. Then we're going to go through the S phase. So this is where we come up with those duplicated chromosomes. Okay, so now chromosome 1 is going to make a copy of itself, both in the egg and the sperm, and then the same thing for chromosome 2. Again, that centromere is going to hold them together, and we have our different sister chromatids. So that was DNA replication. Okay, and then in G2, the main event that's happening in G2 is it's getting ready to divide. And the main thing that's happening is we are getting the formation of our mitotic spindle. So we have these centrosomes that are sending out these microtubules, and they're duplicated and they're separating to opposite sides of the cell. Okay, our chromosome's still duplicated at this point, and again, this is our G2. Right, so now we're actually ready to get into mitosis, and the first part is prophase. So the acronym PMAT, prophase, starting us out there. 3 main events that are happening during prophase. 1, the nucleus breaks down. Okay, so no more nucleus. That means that our chromosomes are going to be in the cell. The chromosomes condense. And that means that they're becoming shorter and thicker. So our chromosome 1, both of those are going to get just a little shorter, a little thicker. Same thing with our chromosome 2s. So they're still duplicated. Again, just trying to make them a little shorter, but also a little thicker. This prevents them from getting all tangled up whenever we start to separate them. And then the mitotic spindle forms. And that's where these centrosomes that were created and separated during G2, the microtubules grow and they attach to sister chromatids. So a microtubule here, remember we talked about microtubules with the cytoskeleton. It's kind of our highway, allows things to move inside. So a microtubule from one side attaches to one sister chromatid. Microtubule from the other side attaches to the other. And that's going to happen for all of our chromosomes. So a microtubule comes and attaches to our sister chromatid here. Microtubule from the other side attaches to the other sister chromatid. This point of attachment is called a kinetochore. And that's on all of the chromosomes. Okay. Our microtubule from the left will attach to one sister chromatid, microtubule from the right to the other, and then our last chromosome down here, again, microtubules from both sides attaching to each sister chromatid. So now we have this mitotic spindle made up of these microtubules formed. We've attached to our sister chromatids, they're condensed, and we don't have a nucleus anymore. Now what happens during metaphase is these microtubules, they're going to play kind of tug of war. They're going to push and pull until these chromosomes line up physically in the middle, so middle of the cell. Okay, so we have our chromosome 1 up here, our chromosome 2. We'll make the microtubules the same color. I don't want to confuse that issue. All right. And our other 2 microtubules, again, we're just playing this tug of war game until we get everybody lined up in the middle of this cell. And there's even what's called a metaphase plate. So everybody's right in the middle of the cell. All right, still attached though. So now in anaphase, what's going to happen is the centromere that's holding these sister chromatids together, it's going to let go. So imagine playing tug of war and somebody lets go of the rope. Of course, you're going to fall backwards. And that's what's going to happen to our microtubules. So then this microtubule shortens, and it's going to bring its sister chromatid with it. The other sister chromatid was attached over on the right, so it's going to separate. And that's going to happen for all of our chromosomes. So now our sister chromatids are no longer attached, but now they have been separated. Okay, so in anaphase, now we separate sister chromatids. A little out of room, so we're going to get rid of this one so that we can move up and now go into telophase. And what's going to happen in telophase, our chromosomes aren't going to be condensed anymore. We've separated them so they can kind of loosen up, but we're going to form a nucleus around both sets of these chromosomes. So at this point, we're going to have 2 nuclei inside of this cell. Again, those chromosomes loosen up a little bit. So now we have 2 nuclei that have formed around both clusters of our chromosomes. And you can see that they are identical to each other. So we still have 2 chromosome number 1s in both, and we have 2 chromosome number 2 in both nuclei. Just like we did at the beginning whenever we started. So we're not changing, we're not separating or like getting rid of any chromosomes. We're literally copying them and then dividing them up. And now this cell is ready to go through cytokinesis to make 2 separate cells. And that's going to be a little bit different whether we're talking about a plant or an animal cell. So in a plant cell, we're physically just going to pinch this cell into 2. But again, each one has a chromosome 1 and a chromosome 2, 1 from the egg and 1 from the sperm. So our homologous chromosomes are still present, but they're no longer duplicated. So we have a chromosome 1 from the egg and a chromosome 1 from our sperm. So that cytokinesis produces those two identical diploid cells. Okay, so we've not changed the genetics. They're the same as each other, and they're the same as the cell we started with. Now in plants, it would be a little bit different. Because they have a cell wall, it would prevent them from pinching apart. So instead, they literally just build a new cell wall in the middle of the cell to divide the cell on the left from the cell on the right. So animals, we pinch them apart using microfilaments, our contraction part of our cytoskeleton. Cells with a cell wall physically build a new cell wall to separate the cytoplasm. In either situation, though, we end up with two identical diploid cells.